Okay, now on to the diffusion of water across the cell membrane, also known as osmosis. Just like in diffusion, we're looking for the area of highest concentration of water to tell us which way the water will move. Now write in your notes that the concentration of water is often dictated by the concentration of solutes in a solution. And that in a solution, solutes and the solvent have an inverse relationship. You remember what the word inverse means. It means opposite. So in other words, when there's a high concentration of solutes in a solution, for example, like if we made a saltwater solution, there would be a high concentration of salt, so we'd call that a salty solution. So when that happens, there's a lower concentration of water, and vice versa. So as we increase the amount of solute in there, the concentration of water decreases. So now we set up what's called a concentration gradient if there's a membrane present. Now, it's also important to remember that water is free to move in and out of the cell membrane with no barriers. In fact, write that down. Sorry, I should have done that the opposite way. But the solvents the water carries often are not. So write in your notes that the permeability of solvents is often limited by their particle size or their polarity. Now, that's not to say they can't get in and out of the cell. But as we've seen in a previous podcast, they're going to need some help. For some, it may be a channel protein. So their particle size may be small enough or large enough that they can't penetrate the membrane, so they need a channel protein. For some particles, their polarity prevents them then from using a channel, so they may have to use an alternate transport method, like, for example, active transport. So sometimes those particles are limited by factors. Okay, so let's continue then. Write in your notes that osmosis is merely the diffusion of water across a semi-permeable membrane. In other words, movement from an area of high concentration to an area of low. And we just discussed how you can have a high and low concentration of water. And you can see now that these tubes are a great example. So you have a high concentration of solute here and a low concentration of solute here. So this selectively permeable membrane then, see the little spaces in the channel or in the membrane? Those particles can't fit in there. So while these particles can't flow between the membrane, water still can, because water is always permeable. So we're going to talk about now what happens in this situation in this podcast, in this part 3 podcast. Now, again, this is a passive transport method, meaning no energy is required in the form of ATP. Let's introduce and review some osmotic concepts and get them in your notes, as they'll be needed in our next lab. Write in your notes that solutions can either be hypotonic, you see these words right here, hypotonic, hypertonic, or they can also be isotonic. So hypotonic, hypertonic, and isotonic. Now also write in your notes that the prefix hypo means under or low. For example, like a hypodermic needle goes underneath the skin. That's why it's called a hypodermic needle. Now, also write that the prefix hyper means above or high, like a hyperactive child has a high level of energy or activity. And then finally, the prefix iso means equal or the same, as in the isometric measurement or an equal measurement. Okay, now let's try and get an understanding of how these terms apply to the environment cells live in. and how that cell reacts when the environment's changed. That will be the focus of our next lab. Okay, so let's talk about why the water moves from high to low concentration in cells. Write in your notes that the direction of movement of water is dictated by the concentration of solute on either side of the membrane. That's extremely important. So it's dictated by the environment the cell's sitting in. This produces three situations you will absolutely be asked about on the AP exam. So they need to be in your notes, and you need to really understand how each is different. All right, write in your notes that in all three cases, it's the concentration of solutes in the solution that surrounds the cell that dictates what happens inside the cell. That's extremely important. Now we're going to look at the effect on both animal and plant cells. So if you look at our slide here, we're going to look at the effect on animal cells, and we're also going to look at the effect on plant cells, because you're going to be asked about either or both on the exam. Okay, first let's look at a cell that's sitting in a hypotonic solution. So we're going to look at this situation first. Hypo. Remember, hypo means low. So write in your notes that since the prefix hypo means under or low, a hypotonic solution has a low concentration of solutes. So if we were talking about it sitting in a saltwater solution, this would have a low concentration of salt, which means a higher concentration of water, correct? Now, also write that if a cell is sitting in a solution that has a low concentration of solutes, water will move into the cell. All right, water will move into the cell because we have a high concentration of water outside and a lower concentration inside. So water will move into the cell. We say that the cell will expand. Now, also write in your notes that if that expansion keeps going, the cell can burst. And that's called lysis, L-Y-S-I-S. The cell can lyse. In fact, if it's a red blood cell, it's called hemolysis that you see here. Now, in a plant cell, it's a good thing. And actually is a good thing in an animal cell unless it's too much. But the cell's pretty good at regulating its own environment. So on a plant cell, certainly water would enter the cell for the same reason. Right? Larger concentration of water on the outside, lower on the inside. Now, what happens now is because this cell has a cell wall, this big vacuole now fills up with water and the cell becomes turgid. So you see this term here, turgidity. So these cells fill up with water, and remember that these cells are stacked on top of each other. So as they all fill up and they're stacked on top of each other, this allows the cell to literally stand up. This happens during the day. During the day, cells take up water because they need it for photosynthesis. That water enters these cells, and the cell stands up and literally is more available to the sun because it also needs the sun to photosynthesize. So, hypotonic, low concentration of solute, high concentration of water on the outside. Water moves into both kinds of cells. All right. If it's uncontrolled in a cell can lyse. It doesn't happen that often. And then a plant cell wants it because it gets turgid, and that cell can then stand up, or the plant can then stand up and photosynthesize more efficiently. Okay, now let's go to the other extreme and talk about hypertonic solutions. So we're over here now at hypertonic solutions. Now write in your notes that the prefix hyper means high or above, so a hypertonic solution has a high concentration of solute. So now we're putting a cell in a salty solution. All right, so in this case, the solution has more solutes therefore a lower concentration of water. So now if we have a lower concentration of water on the outside of the cell than on the in, then water moves out of the cell. And we saw the cell shrinks, which is called crenation. So get that in your notes as well. So in a hypertonic solution, salty water, less water on the outside than on the inside, water moves out of the cell and the cell shrinks. Now the same thing happens to a plant cell now, and this would happen at night. And now what happens is water moves out of the cell and the plant cell becomes flaccid. There's the word, flaccid, F-L-A-C-C-I-D. We would say that the plant wilts at night. So make sure you add that in your notes as well. So when water moves out of this plant cell, the cell does the opposite of standing up, which is wilt. So it becomes flaccid. So hypertonic solution, high concentration of solute, low concentration of water, water moves out, cell shrinks, plant wilts. Now, I think you can predict what will happen in an isotonic solution. So in an isotonic, this is what you strive for, by the way, what your body strives for is an isotonic solution where the cell sits in an environment where there's an equal amount of solutes on the outside and on the inside. So in isotonic, if the cell is sitting in an isotonic solution, water moves in and out at the same rate. It's not that it doesn't move. All right, it moves in and out at the same rate. Remember, water is free to move in and out of the cell whenever it wants to. Now, in plants, same thing. So this is normal turgidity now for plants when water moves in and out of the cell because the cells are in an isotonic solution. Okay. So really the bottom line here is that managing water balance is a key to cell survival. This water management is dependent on water uptake and loss balancing out. It is literally why it's recommended that you drink a gallon of fresh water every day. So in that case, you're striving for homeostasis. Write that down, homeostasis. So water balance is one of the factors in homeostasis. So you need to keep your cell environment isotonic, and to do that, you need to drink at least 64 ounces of water a day. Now, write in your notes that vacuoles are the organelles that help to regulate the amount of water in different cells, and that these contractile vacuoles pump water out of a cell that's getting too full due to sitting in a hypotonic solution. Now, once the cells have enough water in them, these vacuoles do their work to make sure that the cell doesn't lyse. Remember, lyse is burst. So, again, plants have a larger vacuole because water is extremely important in their structure. Your cells still have vacuoles, but not nearly as large as this vacuole you see here in a plant cell. So even single-celled organisms like this Paramecium need this vacuole to regulate water. So in this situation, it's going to use some ATP to pump water out because it needs the energy to do that. So here you see a before and after. You see a full vacuole now. So in order to keep that cell membrane intact, that vacuole squeezes water out of it. Now, back to aquaporins. Write in your notes that it was in 2003 that aquaporins were actually proven to exist in the cell membrane, and that aquaporins allow the flow of water across the cell membrane. So remember, we said that water flows freely in between the membranes. It uses these aquaporins to do it. Okay, that's it for part 3 of cellular transport. Take a picture of those notes, submit them to Moodle, grab that study guide, answer the questions, and I will see you in part 4.